All right, remember with meiosis, the steps will be to go from 2N to N. Or we're going to go from 46 to 23 chromosomes. We got to cut it in half. Or you could also say going diploid to haploid. So let's start with just a nice cell. I'm going to draw big just because I like to do that. Nice big nucleus there. And inside that nucleus should be familiar with the cells you saw last Friday during lab. I'm using purple because I'm going to use pink for girls and blue for boys. And so when you put them together and you can't tell the difference, pink and blue kind of makes purple, right? Well, sort of. So this is the DNA, but it's in its unorganized form. What's DNA called when it's unorganized? Chromatin. This is a cell that's just doing its normal job. It's probably in gap 1, the S, the gap 2. Remember those phases of mitosis? Meiosis is actually going to start out just like mitosis does. Not going to be a lot of difference there. It's just that we're going to have to do a little bit of extra because we have to get back down to the 23. What's chromatin a mixture of? There's DNA in there, but what else is in there? Proteins. Yes, the histone proteins that the DNA wraps around. Yeah. I know. That's for all the people who are gone today. They can have no excuse that they didn't get in on the lecture. Now they'll laugh because I'm talking about them right now. All right, so this cell is in what's called interphase. It is. Black is my kind of neutral color. Yeah, you can't tell my purple up there, can you? No. Bummer. That's all right. Once I get that pink and blue going, you'll be like, whoa, I see that. So remember, interphase is the gap 1 synthesis and the gap 2. All of that is part of a normal cell cycle. Cells making ATP, it's doing its job, all that fun stuff. So that's interphase. And do you remember what the next step was if we were doing mitosis? Yeah, it was prophase was next, and that's the same thing with meiosis, except we're just going to call it prophase 1 instead. And so I'm going to redraw my cell, except, I should have made that more like dotted, because our nucleus is going to start dissolving away, as we know with mitosis, how that happens. Happens the same meiosis as well. And then I'm going to draw me a couple chromosomes here. I'm not going to put them in any particular place, just as long as they're in the nuclear area. Now, what's the form that these are in? Yeah, they're sister chromatids. So the pink one for me is going to be the one from mom, and the blue one's from dad. Keep in mind that there are 22 other pairs going through this. I'm just drawing one pair. If I tried to draw all 23 pairs doing this at once, we'd be here for 2 weeks. So just the one pair is all I'm doing. We'll just say that it's pair number 1. So back here in interphase, the DNA has already been synthesized so that all of these chromosomes can be in sister chromatid form. Now here's something else that has happened that doesn't happen in mitosis. All right, so these are two chromosomes, obviously in their sister chromatid form. During prophase, early prophase, this is the only time that the homologous chromosomes come together. They don't do this during mitosis, but during meiosis, the homologous chromosomes, like the one from mom and the one from dad, come together, and they get so close that they start, well, they call this crossing over, but we can say that they're at a dance and they're grinding right now, and they would probably be getting in trouble, right? So that after they're grinding, look what has happened. Oh my gosh. A piece from dad's has now ended up on mom's. A piece from mom's is now on dad's. And those are corresponding pieces. So it's okay. We didn't lose any genes here because the same amount of genes that were over here have now switched places. So it's equal switching of material. And so what we have done, here's mom's chromosome. Is this sister chromatid the same as this sister chromatid now? No. Remember, this is going to be split apart, these two, because these are two separate chromosomes. But there's a new combination of material on this side than there is on this one. Same as dad's. This sister chromatid's different from that one. And that's called crossing over. And that's one of the things that contributes to our variation. So whenever sperm and egg are made, you never make the same sperm or the same egg because there's so many different possibilities. And at least one spot always crosses over, it seems, on every chromosome. So let's make a little note that crossing over occurs here. Oops, wrong way to spell occur. But I'm not going to draw that on my chromosomes. We'll just take for granted that, yes, it did happen. All right. If this was mitosis, what was the next stage? Metaphase. This is my favorite phase. I don't know why. Well, yeah, I do. It's because you can see it so well under the microscope. So there's no nucleus this time. Oh, this is metaphase 1. No nucleus. That has dissolved away. Our mitotic spindle is set up. I'll draw that after I get my chromosomes in here. And all of the homologous partners, there is none. It's dissolved. All of the homologous partners line up with each other. And I don't want to draw all the other ones, but I will make X's to kind of represent them. Yep, in the middle. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Whoops, I couldn't quite fit 23 pair across the middle, sorry. But it's close enough. The mitotic spindle is connected to each of these. Remember how all that worked? It looked like a spider web. I'm not always going to draw the mitotic spindle. We'll just take for granted that we know it's there and that those are all those proteins from the cytoskeleton and from the centrioles, just like in mitosis, that are going to grab a hold, connect to the kinetochores of the chromosomes, and are going to pull them apart. All right, so that is very recognizable under a microscope. That's why I like that phase. And then we're going to go to our next one, anaphase 1. remember I kind of talked about it like if I could lasso you and pull you towards me, you would kind of, like your belly would come first and your arms and legs would fling behind you. And so that's kind of like what the chromosomes might look like, is their centromere is kind of out front here because they're being pulled by the mitotic spindle. And here's Dad's. Oops, I drew it the wrong way. That's all right. I'll fix it like that. And all the other ones are coming with it. And our cell is now showing evidence that, well, it's going to be dividing soon because our chromosomes have been split apart. The homologous pairs are split apart from one another. We should make a little note while we're on here that, I'll use purple for that, that is a homologous pair. And now they're separated. And on down. Oh. Demanding, I tell you. Ready? No. What's the last phase of mitosis? Telophase. Good. Okay, you may. I may proceed. Wow. I'm going to leave that one there. Don't worry. All right. Well, we know our cell's going to split, so there. That's pretty recognizable. Our cell has almost finished splitting, or the cytokinesis. That cleavage furrow there is almost closed up. Now what can we say about the homologous partners? They are split into separate cells, yes. If this was mitosis, we'd be done. However, this is meiosis. Did we accomplish what I said we would for meiosis? Did we get to 23 or haploid? Oh, we got to do something again because we have 46 chromosomes yet. There's 23 of these things, but they're all doubled. So we're still not there yet because that's technically still 46. And that's too many. Yep, that's why we put a 1 on all of these. So there's a little bit of a rest, or you could kind of call it a little bit of an interphase. But we're going to get started back up again now. I'm going to take that one away. Don't yell at me. Okay. All the beasts are happy out there. Good. The beasts are satisfied. They're not rushing the stage or anything. All right, but now, oh, this will be fun when we draw this stuff. Now we're taking 2 smaller cells along for the ride because we've divided our cytoplasm again. And we're going to just start off again like before, just with 2 cells. This is prophase 2 now. And the nucleus is starting to go away again. And there's Mom's chromosome and there's Dad's. And just like before in prophase, you start to be able to see the chromosomes. They've condensed. No crossing over this time. That's already happened. The homologous partners are split anyway, and that only happens with homologous partners. Those are in separate cells. Metaphase 2. Prophase 2, metaphase 2. Again, here's my favorite phase. Our chromosomes are lined up single now, although they're with their sister chromatid. Because, yeah. So remember the difference last time in metaphase? We had the homologous partners together. Now they're just by themselves. Oh, loneliness. And now we have anaphase 2. I bet you can guess what will happen now. What happened in anaphase 2? Our sister chromatids have split up. And then last but not least, we've got telophase 2. And we know from past experience, our cells need to divide here. Nucleus comes back. What are you talking about me out there for?
No, because I was just confused for a second because they don't look disconnected. I just want something more.
I know.
Basically, she's trying to teach the lessons.
Oh, come on. Yeah. Well, that one kind of is. This one's just farther along. How's that? So this one just has a little cleavage furrow left. That one, that was done. So now we are finally down to our haploid state. Each of these will have 23 chromatids or chromosomes. If we give it a little more time, like 72 days, this might start happening. Yeah, they're like balloons. No, those are sperm. So from one cell, we started with one somatic cell here, or we could say one stem cell. How many products did we get? 4. So from one cell, we got 4 sperm cells here. And they are haploid. And after about 72 days, they will be ready to venture out and try to fertilize an egg, maybe, if they're lucky. Although, with the other millions of competition, who knows who will be there first. Can I take that off? Yep. All right.
Are you drawing for it? Are we done yet?
Nope. if we were making eggs, which, yes, I know, which I love this word, it's called oogenesis. Yeah. Making sperm is called spermatogenesis. Egg making is oogenesis. This obviously would take place in the ovary, the sperm take place in the testes. In oogenesis, the cytoplasm doesn't divide perfectly. In fact, it divides so that one of the products is going to get most of the cytoplasm. So the products will be one nice big juicy egg, and then three duds. These are actually called polar bodies. I call them duds. Correct. They're still haploid and everything, but they're so small and hardly have any cytoplasm or cell organelles that they're just not going to be able to do much. But here we have a nice juicy egg. It's got nice cytoplasm, nice cell parts, organelles, and of course 23 chromosomes in here. It's haploid, and it would be ready to be fertilized for about 24 hours. What's that? The actual, for oogenesis, the actual process for that is halted in kind of a suspended animation. It's halted at about metaphase and doesn't do its final division until after fertilization, which is weird. Which then, as women get older and their mitotic spindles aren't as good as pulling chromosomes apart as they were when they were young, sometimes that last mitotic division after fertilization doesn't get the chromosomes split apart as good as it should, and that's where we get those chromosomal abnormalities we looked at yesterday, such as very commonly is Down syndrome in older women. Although they're starting to look a little bit more at spermatogenesis, that maybe in men that they're doing the same thing as they get older, and their mitotic spindles are leaving extra chromosomes in sperm than what should be. So there's still studies out on that.